structural studies reported in the Zhang et al. Paper was conducted within this building of CNNSI, the California Nanosystems Institute. My lab, the Electron Imaging Center for Nanomachines, or ERCM, studies the structural functional relationships of large molecular complexes, or nanometer scale machineries. Using ERCM's cryo-electron microscopy and high-performance image processing methods, my research team was able to obtain the highest resolution structure in this field. This is the Titan Krios, the latest generation cryo-electron microscope from the FER company. We use it to obtain several atomic models for several large viruses.
In order to keep a biological sample in their native environment and in their metastable state, we freeze sample in vitreous ice under liquid nitrogen temperature. From here, we transfer our sample into cryo-electron microscopy and also maintain the temperature of the sample under liquid nitrogen temperature for data collection. From this remote computer, we control the Titan Krios microscope to take many cryo images of the biological sample. This is a typical cryo-EM image with many identical virus particles. We select and process about 20,000 such particle images to obtain a three-dimensional structure of the virus at a 3.3 angstrom resolution.
All of you already know how envelope virus such as flu virus enter the host cell, but very little is known about the non-envelope virus entry. So this study, we try to answer that question by studying the intermediate subviral particle structure of the acroreovirus, SVP. So shown here is a cryo-EM picture of the SVP. If you average many of those image, and then you'll get a three-dimensional structure as shown here. If I display this structure using a radial color scheme, and you can see how the molecules are put together in making this large SVP particle. What you'll see is that there are many details, including the second structure like alpha helices shown in yellow and beta streams shown in the aquamarine color. And if you cut a piece from this structure, and then you'll see secondary structure, you'll see amino acid densities such as here. And in fact, we see all the 20 kinds of amino acids side chains except glycine. From this kind of density map, we build atomic models of the viral particles. We built 6 different kind of atomic models for the 4 different proteins that are made up of the SVP. Shown here is how these proteins interact with each other in making this large SVP. Most importantly, from this structure, we found that the outermost region of the virus, the VP5 protein, that is undergoing cleavage. The reason for that is that for the virus to enter the cell, you have to remove the protection protein and then leading to the SVP. And if you zoom into the VP5 protein on the outermost region of the SVP, as shown here, we can see that the protein is cleaved at amino acids 42 and 43. Interestingly, we also observe that the fusion finger, as shown here in red, remains buried in the protein after the autocleavage of the protein. So this study demonstrates that the non-envelope virus entry involves multiple processes involving a prime step, SVP, where the protection, the VP5 protein is cleaved, but the fusion finger remains buried within a hydrophobic pocket. From a technical point of view, our study demonstrates that it is now capable to determine structures of proteins at the atomic resolution in the native environment without using crystals. Therefore, cryo-EM should have a broader application in biology for studying structures of many other proteins in their native or even transient stable forms.